All right, in the last podcast, we introduced you to all the parts of translation, of actually building the protein. What we're going to do this time is actually build the protein, okay? We're going to look at exactly how all those parts, the ribosome, the tRNA, the mRNA, how they come together and bring those amino acids into position to become a protein. Just like in transcription, there are three main parts to translation. You start it, you do it, you end it. You initiate, you elongate the strand of protein, and you end it, okay? So, translation, initiation, you have to bring together all the players to start it. You have to initiate by bringing together the mRNA strand with the two ribosomal subunits, the large and the small, and you got to make your mRNA sandwich in between the two ribosome breads. You also have to bring the tRNA carrying the correct amino acid. Okay, so in particular, let's not forget as well, hang on just a second, that in order to initiate, you need the correct amino acid to do that. You need the met amino acid because that is always the first amino acid, because the start codon is always going to be AUG, which tells us we have to put methionine in the first slot. Okay? So, we already said that. So this is looking at what it would look like to initiate translation. You would have the small ribosomal subunit, you'd have the mRNA, and you would have the tRNA would bind its anticodon to that very first start codon, AUG, and it would do this in what is called the P site, the middle location on the ribosome. Now we're going to call that the passive site, and I'll tell you why after we've gone through this a little bit. And you can see we're carrying methionine there. So now we are initiated, we are ready to go. Ribosome subunits in place, mRNA, tRNA with the start amino acid, methionine in place, start codon in place, which is AUG right down here, and the complementary anticodon attached to it. Now, we're ready to elongate, to actually build the long polypeptide, okay? There's three parts to it. We've got to recognize that we've got the right codon-anticodon combination. We've got to recognize that that's in place. We've got to attach the amino acids together once we brought the correct amino acids to the ribosome. And then we've got to shift the ribosome to read the next codon. That's called translocation. Sometimes this whole step is called translocation because we're going to move on down and read the entire RNA strand. So, codon recognition. We are going to read the anticodon. So here we are with our initiation here. Ignore this stuff for a second. And we are at the P site. So now we're going to bring in another tRNA at the A or active site. Except for our first time, that first initial tRNA molecule, every tRNA after that is going to come in and actively, hence active site, match here at the active site. And the tRNA that has previously been matched is just waiting here in the passive site. So the tRNA comes in to the active site, sees if it's a match. Okay? If it is, then we're going to engage in the next step. Okay? If it's not, then it will be discarded. So that is codon recognition happening at the active site on the ribosome right there. Okay? Now, we're going to form a peptide bond, and I'll show you where that is right here. We're going to attach. This guy is hanging out here. This tRNA is waiting with these amino acids attached to him, and he's going to form a covalent bond to this guy right here. So this chain is going to be broken and added to that. Okay? And as you can see, that has happened right here. So now we have amino acid, amino acid, amino acid, amino acid, amino acid hanging out right there. Okay, this has obviously happened many times before. So now this guy is empty, and what's going to happen? Well, he is going to, once we translocate, that means we're going to shift the ribosome to the next codon to be read. This guy is going to get booted out because he's going to get moved to the exit or E site. Okay, so we're now going to translocate. The tRNA is going to now move from the active site to the P site, and the mRNA is going to move along to bring the next codon to the A site to be read. So that's happening here. You can see this shift. The mRNA is going to slide to the next codon to be read. This guy that was in the A site is now shifted to the P site. And then this guy that is now was empty here is shifted to the E site, and he's going to leave. And so now we're just going to do that over and over and over again. We're going to bring in another one at the A site, create the match, and then we're going to go again and again and again until we get to the stop codon where we will stop. Okay? So match, stick the amino acids together, and then shift the RNA so we can read the next codon. Now all this keeps happening until we reach the stop codon, and that would be termination. Stop codons UAA, UAG, UGA. Hopefully no one would expect you to memorize those. At that point, if we reach one of those, a release factor, a protein would bind to one of those and cause the binding activity to stop. We're going to release the polypeptide, and basically the whole assembly is just going to fall apart at that point. So, how does mRNA code for proteins? Let's just have a little review. If this is your DNA molecule here, you would, to create mRNA, you would go complementary to T, which is A, complementary to A, which is U, complementary to C, which is G, and so on down the line to make an mRNA molecule. And then to make a protein, you would read AUG, look at your genetic code chart, see that that codes for methionine. Look at CGU. It's easiest if you divide this into threes. Find the start and then divide into threes. Okay? And then you'd find that this codes for arginine, this codes for valine. Of course, you'd have to have your genetic code chart, know that, and so on. And that's how you would get your protein built. Okay? So, I think this is just showing us what I just showed you, so that's nothing new. This is the codon, and that is what is in your genetic code chart. Always it is codons on the mRNA, not the anticodons. Those are not represented in the code chart, so don't twist yourself up in that way. But if you were a cell, you wouldn't have a genetic code chart, so you would have to use the anticodon of the tRNA to help you know that MET was the match for AUG. Okay? So that's kind of how that would work if you're really a cell. So this is a really nice diagram showing you that you've got the DNA making RNA by using an enzyme called RNA polymerase that's shown here. We're going to make RNA, but that RNA has got to get processed. Remember, it's got to get processed. The exons have to be kept and the introns are cut out, and so we edit that RNA through RNA processing. We put the 5' cap and the poly A tail on it to protect it, and then we send that mature RNA out to meet the ribosome, where it's threaded through the ribosome, and then we are going to go through initiation, elongation, and termination with the tRNA molecules, and bringing the amino acids to that ribosome and making the polypeptide. Okay? It's also important to remember that that polypeptide, that protein that is made, is not necessarily finished. It's going to start to fold in a shape to create a specific conformation or a specific shape. And of course, we know shape is really important. The gene sequence determines the order of amino acids or the primary structure. And then that there are some helper or chaperone proteins that can kind of help it fold, if you will, in correct ways sometimes. We can modify it, we can add amino acids or alter them or remove them altogether. The polypeptide may be cut up, so there's all kinds of ways to modify it after it's made. We can even put 2 or more polypeptides together. So by no means is that the end of the road right after translation. This is reminding you of primary, secondary, tertiary, and then quaternary structure, where we fold that protein, and if you don't remember that, you might need to go back to your biochemistry unit. All right, that's it for this podcast. I will see you next time.